Hello everyone, welcome to part 2 of my Aducanumab series on the biologic developed by Biogen. So in the previous video we talked about pathogenesis of amyloid beta and the amyloidosis theory of Alzheimer's disease and the link amyloid beta is proposed to have with neural degeneration. We also talked about Aducanumab, the method of action, the type of drug, a bit about the structure, a bit about its effects and its properties. So this video, as promised, we're going to talk a bit more about the context surrounding the drug in the field of Alzheimer's biologics that target amyloid beta proteins, and a few more things about the drug which I mentioned in the last video were a bit controversial, including most notably its rejection of approval by the EMA. So yeah, much to be looking forward to. We're going to be looking at many multitude of different sources. We're going to be looking at other drugs of a similar drug class. And in fact, we're going to be looking at the amyloid beta hypothesis itself rather critically, because recent evidence of the past few years seems to undermine this theory. So a lot of interesting stuff, and hopefully you'll stick with me and enjoy this video. Thank you very much. D.Knopman team's review on Aducanumab and Biogen's decision to take it forwards after the futility declaration in 2019 from the Emerge and Engage trials. So it's interesting that David Knopman described the trial landscape for these neurologics, Aducanumab, gantenerumab, solanimab, and a few others, which we'll get into later, as bleak, which is the title of this section. Now, since 1999, with D. Schenk's study showing that immunization against amyloid beta formation into protofibrils and oligomers and so on, decreased the deposition of these amyloid beta and was linked to neurological benefits for mice. Ever since then, it's been a bit like a gold rush, and several companies such as Eli Lilly, Biogen, and others have been looking to develop a neurologic for Alzheimer's, which would be the first treatment for Alzheimer's with efficacy to drastically slow the development of the disease, the onset of symptoms, and even some might have hoped to reduce and mitigate the disease itself. So, with such high hopes at the turn of this century, well, what is there to show? Well, so far, apart from perhaps promising results from lecanemab and the questionable results from aducanumab, very little has been shown in terms of benefit. All of the trials so far, most notably solanimab, have been halted for futility. Earlier on, many were stopped because failures to even reduce the amyloid beta that was being linked to pathogenesis. And later on with aducanumab, perhaps more discouragingly, with futility was put in because these drugs were not actually showing a great deal of clinical benefit. So, for example, the aducanumab trial, multiple Alzheimer's tests and brain cognition tests, which we'll get more into later, were taken, and in fact, apart from very slight improvements in the Emerge trial, very little was actually reported in the way of significant benefits, and that's in the slowing of disease onsets compared to the placebo group. So that's quite concerning. A lot of money has been put into this field, and companies certainly don't want to waste the millions, if not billions, of dollars. But yeah, there's a lot of failure in this particular field, and that's what made Biogen's decision to submit aducanumab to FDA approval, even though the ENGAGE trial had failed to meet clinical endpoints, and the company specifically set a goal that both the EMERGE and ENGAGE trials needed to meet expected endpoints, otherwise futility would be declared. They did a post-hoc analysis in 2019 and then decided they would submit to FDA approval later. And that's what prompted D.Knopman and team to do a review on aducanumab and why Biogen thought this was appropriate. And obviously, if one trial, EMERGE, is showing statistically significant benefit between high-dose aducanumab and placebo of the slowing of symptomatic onset, but your ENGAGE trial is showing no real improvement whatsoever, the improvement was very low. In fact, for one of the trials, there seemed to be a very minor, not statistically significant, but decrease in the slowing of onset of symptoms as opposed to the placebo group. So that's a very concerning result. So you've got a null result and you've got a positive result, but a weakly positive result. And D.Knopman argues that, well, the former could be just as likely true as the latter, even with a post-hoc analysis done, which Biogen claims justified submitting aducanumab for FDA approval. Now, of course, we all know that aducanumab is now FDA approved, though it was not EMA approved, which we'll get into more later. But now we're going to do a little detour to further explore the idea of the field of aducanumab and the neurologics that purport to treat amyloidosis and thus Alzheimer's disease by having a little case study of solanzumab, which is a drug developed prior. So, solanzumab. So solanzumab is a biologic developed by Eli Lilly for the express purpose of treating Alzheimer's disease built upon the amyloid beta hypothesis. So, in 2016, the phase 3 clinical trial for solanzumab was completed, and the results were disappointing. There was very little reduction in amyloid beta deposit, and there was no clinical benefit showed, as outlined in Eli Lilly's update. So solanzumab was specifically designed to target patients with Alzheimer's disease that had amyloid beta deposits in early stages. That's hypothesized to be the group of people that are best treated by this drug. So its failure was pretty significant, especially since this was a phase 3 trial. So in an article by the Alzheimer's Drug Discovery Foundation, it outlines the disappointment and failure of solanzumab, and actually talks about its decision to stop offering funding for drugs targeting beta amyloid, since the foundation felt that too many drugs were being pursued in this category and with so many failures, they were beginning to wonder if the money was being well spent. All said and done, solanzumab was an early failure, but in the timeline of aducanumab and the newer drug lecanemab, but still a significant one, and one that had more ramifications on the field. So now we're going to talk briefly on the amyloidosis hypothesis itself and bring forth some of the challenges that have been made against it in recent years. So this isn't directly, well, it is directly relevant to the video matter, but ultimately the question will not be answered by me in any full way. I'm just going to present the hypothesis and have it as an interesting nugget of information before we get into the meat of the criticism and perhaps defense of aducanumab as a drug on its own. But regardless, amyloidosis as the foundation, Amyloid beta cascade, as the foundation for clinical efficacy for this drug, aducanumab, it's indeed significant if this hypothesis is correct. So first I'm going to look at an article in Medical News Today which proposes the idea of the amyloid beta hypothesis perhaps not being correct. Significantly, Dr. Anton Porstensen, who did post-hoc analysis on several influential amyloid beta trials, outlines his opinion that it's not the formation of amyloid beta plaques from loose soluble amyloid beta protein. It's not the plaque formation, so plaques, deposits, fibrils, etc. But actually it's the loss of amyloid beta 42 from its regular function. So his exact statement is higher levels of soluble amyloid beta 42 is therapeutic, and it's in fact the soluble 42 residue proteins being caught up in the plaques, the fact that they're no longer free in the cells and in the brain is actually the culprit for degeneration. Now, this is not a substantiated claim as put forward by Dr. Rebecca Endelmeyer from the Alzheimer's Association, but it is indeed an interesting hypothesis, especially since so much of what we would expect from drug trials if the amyloidosis hypothesis were indeed correct has been conflicting. And on that note, I'm going to get onto the drug Verubecestat, which is a drug developed by the German pharmaceutical firm Merck. Now, this drug is a BACE inhibitor, specifically BACE1, which is a amyloid precursor protein cleaving enzyme. So, we looked at amyloid beta precursor protein last video, and the idea behind this drug is if the enzyme that cleaves amyloid beta precursor protein is being inhibited, then amyloid beta will not be produced because it's not being cleaved, and thus there'll be no chance for plaque formation or fibril formation or any of these other harmful mechanisms of action of the amyloidosis disease. So clinical trials should show that there is a massive reduction in symptomatic onset if the hypothesis is true. And this is very significant, even more so than the other biologics previously tested, because while Aducanumab targets amyloid beta plaques and other drugs, Solanezumab targets already free-floating soluble amyloid beta 42, this drug actually prevents its formation. So if anything is going to provide an efficacy claim for the amyloid beta hypothesis, it will be Verubecestat. However, Verubecestat was terminated for futility after the clinical trial reached a certain point. So 1454 patients were enrolled, 485 received a 12 milligram dose, and 484 received a 40 milligram dose, and the rest received placebo. So I'm going to go through the results now. There was actually a worse outcome for the higher dose group than the placebo group. So the CDR-SB, which is a cognition test score, was 1.65 in the 12 milligram group, 2.02 in the 40 milligram group, and 1.58 in the placebo group. So that is actually a worse outcome. If you want to look up the CDR-SB test, please do so. I'll provide some links. So that's a very interesting result. A drug that attacks the source of the problem directly, the formation of soluble amyloid beta, which could form plaques, could form fibrils, with free-floating, that actually induces worse outcomes. And these are statistically significant outcomes with a p-value of 0.01 for the comparison between the 40 milligram and the placebo group. And that's especially significant as Aducanumab purports to treat only with a higher dose of Aducanumab, as the lower dose showed no statistically significant efficacy. So it's significant to note that this higher dose Verubecestat had a worse outcome for patients rather than placebo. And this cannot map directly onto Aducanumab, as amyloid plaques are different from free-floating soluble amyloid beta. It may indeed be possible that amyloid plaques have a negative effect on neurological function for cells affected, even while amyloid beta free-floating has a positive effect according to this counter-hypothesis on amyloid beta's role in Alzheimer's disease. All that being said, it's very interesting. Time will tell if the amyloidosis theory is indeed counteracted or whether it is verified. There is, especially with the approval of Aducanumab in the recent years, there is indeed a scientific debate going on now, which it will be interesting to know the results of. Perhaps in a few months or years, whenever some significant data comes out, I'll be able to revisit this topic, and yeah, that'll be very interesting. But this is just a bit of a food for thought section, and hopefully provide a little bit more context and information on the drug in this field. Thank you.
We will have literally stars before midnight tonight. This is a breathtaking page.
In this section of the video, I'm going to be taking a look at Aducanumab's eMERGE and ENGAGE trials and some of the details and information, and specifically the results that accompany. So, the ENGAGE and eMERGE trials. And these are the phase 3 clinical trials which were first put in place in order to study Aducanumab and its effects. Right. So, here's a bit of information on the patients. So you've got 2 identical 18-month randomized, double-blind, placebo-controlled phase 2 studies. So they're pretty valid. They've got pretty good controls. 3,285 patients. That's pretty. It's a good sample size. It's enough to have a fairly good idea that your data is consistent and can be applied in theory to a large population, especially since it's using 348 sites in 20 countries. Though, as you can see, these countries do seem to most heavily impact European and European descent nations, which will be of relevance later. That being said, the primary endpoint is described as the CDR-SB test measuring cognitive decline at 18 months. And there's 2 dosing regimens, low and high, which is compared to placebo. So these were these are some of the very basic parameters of the test. And it's also got some other endpoints which are secondary, like the MMSE test, ADAS-Cog tests, a couple others, and of course the sub-studies which are determining efficacy of the reduction in amyloid using PET scans, the reduction in tau fibrils, and other disease-related biomarkers. So all in all, a fairly complex study, which it's relevant to note would have been very expensive. So, dose regimen, and here are the top-line results, which are the important aspect of this. So for the eMERGE trial on the left and the ENGAGE trial on the right, remember that the eMERGE trial was deemed successful and the ENGAGE trial was not. So, we've got the demographics. So, we've got patient deposition. Here at week 78, we have our primary and secondary endpoints. So that's a year and 26 months in, 26 weeks. Let's try again. So in this section of the video, we're going to be looking at the eMERGE and ENGAGE trial results and some details of them specifically and directly from the Aducanumab report by Biogen. So yeah, let's get into it. So here's some very basic information on the eMERGE and ENGAGE phase 3 clinical trials. So you've got two identical 18-month randomized double-blind placebo-controlled phase 3 studies. Now it would end up going on for 72 weeks, not the full 18 months, as the trials would be ended for futility, as earlier mentioned. But yeah, randomized double-blind placebo-controlled, these are fairly extensive measures and should lend some validity to the results of these trials. 3,285 patients over 20 countries and 348 sites. So those numbers are pretty good. You can have a fair degree of confidence with such that these results can be applied to a fairly broad population, at least in theory. So that's pretty good. It is important to note here, and we'll get into this later, that these are very Western European countries by descent and by nature and ethnicity. It would be mostly white and white ethnic, so that is important, as it's not fully representative, perhaps, of the populations which may be treated by this drug or may already have been treated by this drug. But yeah. 2 dosing regimens, low and high, and placebo will be the third group for these patients, which will be divided between the 2 trials, as seen below. The primary endpoint set by Biogen was the CDR-SB test, which is a measure of cognitive decline, and with other endpoints such as the MOC test, the ADAS-Cog test, and of course sub-studies on amyloid beta, tau detected in PET scans, and other related biomarkers. Right, so here's the map with the countries. So yeah, mostly very heavily weighted, as you can see here, towards Asian and white ethnicities, which will be, especially for America, which will be relevant later. So dose regimen, we've got the baseline demographics. So indeed, we can see in the eMERGE and in the ENGAGE, you've got a fairly good amount of, a fairly representative amount of women compared to men. You've got a pretty big representation of Asian and white ethnicities. You've got a fairly good representation of education years, so that's pretty consistent across the entire data sets, and 14.5 years. Again, this is more specifically applicable to perhaps we could say higher income countries, between 14 and 15 years of education, but again, not too relevant, other than interpreting the results of the cognitive tests. We've got information on medications, ApoE4, which is relevant because this gene has been linked to amyloidosis, and it's also been linked to amyloid-related imaging abnormalities and complications caused by these neurologics, which we'll get into later. This is a bit bigger topic on maleficence that we'll address. Right, so baseline disease characteristics. So this would be just the initial results, I suppose. Got patient depositions here. So So completed placebo-controlled period. So in the eMERGE trials, the ENGAGE trials, you can see the number of patients here. So this is comparing the patients, looking at the patients that discontinued. So, adverse event, consent withdrawn, death, so on and so forth. So yeah, looking at how the population was affected over the 72 weeks that this trial took place. Pre-specified primary and secondary endpoints at week 78. So here we've got some results. The Emerge trial, which I said previously was the more successful trial. So here we have for the primary endpoint CDR-SB test, we have a pretty good reduction compared to the placebo group in progression of cognitive impairment. So minus 0.38, which is statistically significant with a p-value of 0.012, as opposed to 0.09, which for a still fairly impressive reduction is not statistically significant in the low-dose aducanumab. MMSE, we have a similar, also reaching the critical value as for the ADAS-COG test, and also for ADCS-ADL-MCI test. So these are some pretty good results. So you can see why Biogen would bring forward these results after their post-hoc analysis. It's fairly promising. Again, this is all the slowing of onset of symptoms. So this is, how much worse placebo got as opposed to those with high dose. So this is far from a cure, but still, if these were the only results that existed and they were duplicated or similarly shown for the Engage trial, there wouldn't really be that much controversy because they're pretty good results. While there would perhaps still be controversy on the matter of abnormalities and edema, which we'll get into later, but no, these results seem to show clinical efficacy, but there's more to that story which we'll go over later. Now, the Engaged results. Now, I mentioned earlier that they appeared to be not as statistically significant, but an actual decrease in outcomes for high-dose aducanumab in one of the trials, and this is it. 0.03, which means that the CDR-SB test displayed actually a very tiny, so probably not significant, but a very tiny worsening of outcomes for patients who took the high dose of aducanumab. So that's pretty significant. Certainly you wouldn't be able to argue much better than a null result from that. But again, this is just within this specific trial. No statistical significance in any of these tests, though in the ADCS-ADL-MCI test there's a reasonable reduction, but still, as there was in the high dose, though of course the high dose in Emerge was statistically significant, whereas this wasn't. So really, this is a very different set of results, and it's interesting that this comes about because, again, if we looked up again at the demographics and the patient distributions, there would be no real, there'd be no clear and obvious reasons why such drastic differences would be seen between the two groups. Though, interestingly to note, placebo decline in the Emerge trial was far, far greater than in the Engage trial. So 1.74 to 1.56. That's a big difference. And as we mentioned earlier, D.Knopman seems to think that this provides some questions as to how much of the improvement compared to the placebo and the aducanumab dosing can be directly attributed to the drug, and how much can be attributed perhaps to a placebo group, and specifically this placebo group performing worse than expected. So again, these are fairly similar results, these are fairly similar, this is perhaps slightly worse, but this is a far worse result, and of course this is the primary endpoint, so it's most significant. Yeah, here's some longitudinal changes in baseline. So this is just the results shown in graphical form. So this is how markers of cognitive decline, and this is showing the cognitive decline getting worse. But of course, in the Emerge trial, it shows that high-dose aducanumab, shown by the purple, there's significant decrease in the symptomatic biomarkers, and statistically significant, as shown by the star represented here, compared to the placebo. But here with the Engage, this isn't seen. And for a final point, which is very significant, especially compared to the Solanezumab trial, very, very statistically significant. So I think this represents 3 standard deviations. Well, 3 ranks of significance. Really high reductions in amyloid, as shown by the PET scan, especially for the high dose of Aducanumab in both trials, Engage and Emerge. The very slightly very slightly lowered reduction is in the Engage compared to the Emerge trial, but certainly not enough to really speculate over. Now this is really interesting, because this drug has actually shown efficacy in its the surrogate marker for Alzheimer's disease, and efficacy on paper what it's supposed to do. It's supposed to reduce plaques, which would reduce the amyloid in the showing up on the PET scans, and it's done that. It's done that very remarkably well. Again, compared to Solanezumab or perhaps other drugs, so it really has met its expectations with the surrogate marker of amyloid beta plaques. But again, we can see here cognitive tests. We're not seeing, even in the positive result, we're not seeing anywhere near the corresponding reduction in pathogenesis of Alzheimer's disease as a result of reduction in amyloid beta. So keep in mind the earlier data and queries we looked at of the amyloidosis hypothesis. Again, that's a topic for another time perhaps, but just interesting to keep in note. Tau, we've got tau here. Again, we're showing a good reduction in p-tau. Also a reduction in total tau, which is perhaps not the best because, as mentioned in the previous video, tau is an important protein for the organization of the protein microtubule highways. But yeah, still very interesting stuff for both the Engage and Emerge trials. So showing the reductions of the surrogate biomarkers that were necessary for the clinical trial. But I think that's enough of a look at the Engage and Emerge trials. Again, I'll be linking the data in the video, which is directly from Biogen in a PowerPoint presentation to its investors. So yeah, some food for thought there. Right, let's get into it. So here is some preamble to the recommendations and a specific analysis. So here it does talk about the Schenk et al. Paper, this bleak trial landscape, which is the phrase, of course, I magpied for a previous section because I found it pretty useful. Yeah, it talks here about perhaps the unfortunate reality of this landscape and how it was disappointing but not unexpected that Aducanumab would have utility, but 7 months later, claiming efficacy after a post-hoc analysis. This is the unexpected aspect of the story, which is what this article is questioning, and David Knopman and team. So here they're putting out there they don't agree with the claims for efficacy on ADU. So they're going to forego a discussion on the magnitude of the claimed efficacy signal for ADU. Again, because amyloid beta, clearly there was a really good response in amyloid beta reduction. And if that was a surrogate marker that mapped onto clinical efficacy, that mapped onto reduction in decline, then all well and good. But you can't. But if one says one thing, so if one's going down and the other is not following, then you can't really use that in a discussion about the actual efficacy of the drug, because the drug's treating Alzheimer's, it's not treating amyloid beta buildup. mentions here some of the ethical and justice and malfeasance concerns, which we're going to go into later on the AN position statement as in a separate article. So yeah, here it talks about the phase 1 trials, which interestingly enough talk about the efficacy of aducanumab in reduction of amyloid beta. So it was efficacious in the surrogate marker throughout the trials. Here mentioning the futility announcement in March. Yeah, disappointing day, and of course Alzheimer's is a significant problem in the West, especially, but around the world, and several organizations and families and those affected really have a vested interest in this treatment's arising, especially since it's a rather, in fairness, as many neurological conditions and psychiatric conditions have been similarly, it's a very barren field, and that's disappointing indeed. So yeah, October 2019, so 7 months later, there's the press release, C-Box, where Aduhelm is described as efficacious. So they did a selective view, so your post hoc analysis, and so they made an efficacy claim. So they would have looked specifically at the EMERGE trial, which we looked at earlier. So again, there were statistically significant benefits in the test, measuring cognitive decline, the CDR-SB test. So indeed, there is some basis for that claim. So here they make their disclaimer, Knopman et al., that their analysis is limited by a lack of peer-reviewed publication and their reliance on the data selected by the sponsor. So they've applauded Biogen for presenting a lot of data, some unanswered questions, missing subgroup analysis. They're just laying out all the grounds of their evidentiary basis. So yeah, let's get into it. So EMERGE trending positive, engage, and likely to meet primary endpoints, which again, both trials had to meet primary endpoints stated by Biogen of these clinical trials in order for them to actually continue. And again, after 78 weeks, cut for futility. So that's how that went, obviously. So they do, Knopman does mention that, the trends that we saw in the data by 78 weeks wouldn't necessarily have continued mapped onto the entirety of the 18-month period. So you would have to make another step to grant that EMERGE would have continued to trend positive, though not too much of a leap there, considering the positive trend was consistent throughout the 78-week period. But they do point out the point I made earlier, which is that if you've got a positive in EMERGE and you've got a negative in ENGAGE, two trials both have good population distributions, equal representation of high dose, low dose, and placebo, good representations of women and the two ethnicities, primary ethnicities, which were measured in these trials, white and Asian. So that being said, both trials should most likely going to be as valid as each other in comparison, and one's giving you a null result more or less, and one's giving you a positive result, but a weakly positive result in the grand scheme of things. So David Knopman points out that either just by basic logic, either could be as likely to be true as the other, as likely to be representative of the condition and the effects of aducanumab as the other. Right. So yeah, it talks about the post hoc analysis here, not equivalent to predictive power to primary pre-specified analysis. So he goes through this here. variations in execution that introduced heterogeneity. So this is the important part of the post-hoc analysis, because Biogen made the claim that because there was a greater exposure to high-dose aducanumab in the eMERGE trial, well, that's the actual reason why the eMERGE trial had better results than the ENGAGE, simply because, as it outlined here, there was a difference in the way the doses were given to patients. So that's the assertion that Biogen have made from their data. And this is, of course, the critical variable that prompted them to submit an FDA approved for FDA approval. And he mentions, as we saw earlier, the placebo difference, so 1.74 versus 1.58, if I recall correctly, which is a significant difference in decline on the CDR-SB test. So let's see here. Cause it perplexing, and again, I pointed out there wasn't really any real reason why this should happen. But I mean, if you've got unknowns, if you've got something that you can't quite understand, all the more reason to want to run another clinical trial. That's not something that goes in Biogen's favor, specifically. So he's got a condensed version of the results we saw earlier. Right, so talks about the biomarker data, tau data. Right. So here he's going to make his, he's making a value judgment on Biogen's claim for efficacy and saying, well, I don't think you can make a claim for efficacy even with your post-hoc analysis. As you know, Biogen asserted that, well, it was the fact that there was more high-dose aducanumab, more consistent dosage in the eMERGE trial. That's probably why there was more better outcomes. But a counter hypothesis, which is proposed by Knopman, is that, well, you've got errors, not errors, you've got significant deviance with the placebo groups of both of the two trials, with the eMERGE, which was, of course, the one trended positive, their placebo group declining far faster than that for ENGAGE. So he purports that, well, perhaps there was a problem with the placebo group in the eMERGE trial as opposed to positive, which would have more favorably compared the placebo group to the high-dose group, as opposed to more efficacy for the high-dose group. So and then he makes the judgment based on his judgment, based on the data, that it doesn't support the conclusion of clinical benefit, which is a fair conclusion to draw holistically from the study. And then, suggesting that if the analysis suggests there's an efficacy signal definitively, then that's unreliable. But there is a legitimate rationale for undertaking a definitive third phase 3 trial, optimally designed, adequately powered to prove efficacy. So he's saying, recommend a third trial before you go through with this and before you actually propose to FDA. Optimized and adequately powered. Well, the previous trials were pretty valid, optimized, and highly powered as clinical trials go. So this is not an attractive proposition for a multi-billion-dollar company. Certainly not very profitable, especially if, and now with data coming out about the amyloidosis hypothesis, especially if they feel there's no chance of a third trial actually showing clinical benefit. So that would be a money hole, potentially, and no investors going to want to get behind that. So while it's a clinically prudent decision, not a financially prudent one perhaps, and that is important, because if there's no money to produce more drugs, then all the possible benefit brought about by high standards and good ethics just cease to exist. There's no we can debate ethics and standards all day and all night, but if there's no money and there's no impetus to create drugs, then it's just talk and then nothing actually happens. So economic and financial concerns are indeed important. All right. So now we're going to get into the safety concerns, which I mentioned earlier, which will form a more complete picture of aducanumab as a drug which should or should not be FDA approved. So D.Knopman and previous bits of information articles we've looked at have mentioned safety concerns with this neurologic aducanumab, which as a matter of fact, are safety concerns which are fairly unilateral across the field of neurologics which target amyloid beta plaques or similar deposits. So amyloid-related imaging abnormalities are, of course, the most prominent of these, which we're going to look at here in Biogen's results, and then we're going to look at a definition of AR or IH, which I have here, and we're going to look at later the AAN position statement, which more clearly details problems with this with these side effects. But it's important to note that for amyloid-related imaging abnormalities, several patients were discontinued. Some were removed from higher dose to lower dose during this study. So this was a fairly significant event. And most significantly, a patient, if I can pull up the article here, Reuters, a patient was killed, well, died, and it's possible they were linked to IIH because it was a brain swelling edema which led to the death. So this is the Reuters article here, but we'll get back to that in a minute. So the safety summary here, we've got patients who had AE, so let's see, AE is adverse event. So patients who had an adverse event, so that's a fairly high number of the patients. As a percentage, we've got between 80 and 90%, but 95%. But again, adverse events can mean a lot of things. Headaches and stuff certainly could be linked to the disease itself rather than necessarily, or some aspect of the treatment rather than necessarily caused directly by the effects of the drug. But again, 90% and a consistent 90% across all groups is pretty significant. It does seem, if it's similar from placebo to high dose, it would seem to exonerate the drug from culpability with these events in particular. Patients with significant adverse events, again, similarly, it's about the same across the emergent engaged trials for placebo, high dose and low dose, which again seems to exonerate aducanumab from culpability here. Now, patients permanently discontinuing treatment due to an adverse event. We've got differences here, but of course, there's a difference in a patient discontinuing treatment they're actually receiving with an adverse event as opposed to patients that are on placebo. But this is the interesting statistic here. Patients permanently discontinuing treatment due to amyloid-related imaging abnormalities, which of course have been linked to aducanumab usage and usage of similar neurologics. So massive increases across both trials, which are consistent for both the emergent engage, despite their differences in clinical efficacy. So this is pretty significant. Amyloid-related imaging abnormalities, again, have been linked to pretty serious side effects. But yeah, we'll go into all this later. Adverse events with incidence. So ARIA E and ARIA H. So this is microhemorrhage. Again, very large increase for high-dose aducanumab over both. That's very statistically significant. And a microhemorrhage is nothing is a very serious matter. Superficial siderosis, again, mapping here, very similar, big increase, especially with the higher dose. So as we can see from these results, a higher dose is indeed linked to a worse outcome when it comes to these side effects. Headache doesn't appear to be, might be statistically significant, not as significant as the increases in ARIA-E and ARIA-H incidence. Patients with any event, that's a fairly significant increased adverse events with incidence, so that's something that actually is a cause for concern. So again, these are significant increases, though again concentrated over ARIA-E, ARIA-H. Fall pretty much the same, but that would seem to indicate there's not a real benefit if falling is associated with Alzheimer's disease, and falling incidence is around the same for patients if they're taking aducanumab or not. That's one marker you would hope if the treatment was efficacious, one marker which would indicate that the treatment is working if falling incidence was less for higher and lower dose aducanumab as opposed to placebo, but not shown by the data, which is unfortunate. Indeed, an increase, though falling is a bit more multifaceted and something we can specifically put on a drug, but an increase in engage. So that's just something to note. ARIA incidence, again, big increases with high dose. Interestingly enough, ApoE4, which is a gene again which we linked to amyloidosis, much higher incidence for ApoE4 gene carriers, which is why, if I recall correctly, the drug has on its label has a very specific caution for patients who have the ApoE4 gene in taking it. I believe there is a mandatory test as part of the treatment proposal for an individual patient, at least in the United States, which is where it's actually approved. Right. So that's Biogen's data on the subject. I'll just look here at the Reuters article, which it's very brief. It just notes that very brief, and just mentions the death of and then it talks a little bit more about aducanumab. So Roblox Hospital Insurers, that was a separate article, and then here's the patient who died with brain swelling. So if we go on to Frontier's article on amyloid-related imaging abnormalities with anti-amyloid antibodies, which, as we talked about extensively last video, aducanumab is one, and all of these neurologics are. Talks about the second generation anti-amyloid monoclonal antibodies, but the most common side effects are ARIA, and this is a significant risk as described by the article. Meta-analysis, so donanemab and aducanumab will be mentioned somewhere else here. Aduhelm, lecanemab and gantenerumab. Headache, worsening confusion, dizziness, visual disturbances, nausea, and seizures. These are important. It's significant that it's these specific symptoms, because many of these symptoms can be linked to progression of Alzheimer's disease. ARIA-E, linked to excessive neuroinflammation. So these are all bits of information which are kind of peripheral to the main thrust, but I think the main point of this section is ARIA-E, ARIA-H. These are significant side effects which are directly associated with aducanumab, associated with the other neurologics of the class. But this is an interesting point to be made, and the ramifications of these side effects, we will look at in response to the AAN article, which my commentary will provide most of the thrust for criticism of Aducanumab and the FDA's decision to approve the drug. But yeah, that'll wrap up this section. And so we're going to get on to the AAN position statement and then talk about a little bit about the EMA's decision to reject Aduhelm. All right, so published in Neurology, this is the AAN's position statement. Right, so yeah, let's get into this. So it talks about Alzheimer's disease, just a brief overview of the condition. FDA approval, this will be relevant later. And it begins to outline its arguments that there's lowering standards of scientific evidence used for drug approvals, which is a bit of an accusation, but again, we'll get into that. Aducanumab, which is of course the subject of this article. So they mentioned some interesting bits of information here. So drug approval traditionally required 2 well-conducted clinical trials with positive results. But of course, Aducanumab, the trials were stopped for futility. Now, to say both were stopped for futility, 2 studies stopped prematurely for futility is perhaps a bit misleading of a representation, because one of the trials had that positive outcome and one of them had, of course, a negative outcome. But I mean, regardless, both of them were stopped for futility because they didn't meet the endpoints. So it's a very valid concern that there was one standard for the FDA, which was not carried out in Aducanumab. So an advisory committee, which was commissioned by the FDA, 10 votes said that Aducanumab, the data doesn't show that Aducanumab shows a slowing cognitive decline. So that's not even a decision on whether Aducanumab should be put into the market or not, because that decision would of course be weighed by financial incentives, which will be got into later in this article. It will be weighted by the concerns of amyloid related imaging abnormalities and edema, which again will be gone into later in this article. But just a decision on clinical efficacy, you've got 10 experts saying, and this is the panel the FDA brought forward, 10 saying it doesn't show convincing evidence, which is again a fair assessment of the data, as we looked at earlier, and 1 saying uncertain. So that's not a single positive vote. And keep in mind, the FDA did indeed approve this drug later. So this is interesting. So the accelerated approval pathway was invoked, which is uncharacteristic. This is the sort of measure you might expect for a vaccine, perhaps, where there's immediate and significant interest in getting this drug out as early as possible. Now, there is a very prominent financial interest for Biogen to have the drug out as early as possible, because they do not want to run another clinical trial, as we looked at earlier. But there's no, from a public health perspective, of American public health, and of an expert clinical perspective, from perhaps the position of the FDA's team and that was commissioned, there's no reason to get, to rush development of this drug. That's not how Alzheimer's and this field works. And so they base their decision on the surrogate marker, which this article doesn't like, and is fair, because the surrogate marker if it can't be linked directly to clinical efficacy, then it's not really a surrogate marker. If you can't link the reduction in amyloid beta to an improvement in clinical outcomes, then what use is that marker, which this article asserts that the FDA used to approve. And yeah, it talks about that reasoning there. 3 members of the advisory committee have since resigned. Now, I mean, I'm not super familiar with the politics and the precedents of this kind of event in an American healthcare and regulatory system. For all I know, this is something that could be relatively common, something that could be very rare. So I'm not going to comment specifically on that, but it is interesting to note that 3 resignations of this panel, it's not something that you would want. That's your first ethical concern. Why on earth would the FDA approve this thing? It's odd. I mean, especially when the far more reasonable, from the regulation committee's perspective, the far more reasonable approach should have been should have not been approval or denial. It should have been, you need to run another phase 3 clinical trial before we can be satisfied in the efficacy of this drug, which again is what David Knopman recommended and his team. So another ethical concern, the cost. $56,000 yearly cost. That's a high cost for a drug, especially a new drug. And of course it has to be. They've put a lot of money into this drug. It's probably going to be fairly expensive to produce as a biologic. And they're going to have to recoup all the costs for the clinical trials, which would have been very expensive, as we looked at earlier. But that is a very high cost. That's above the medium, if my memory serves, that's above the medium income of Americans. So yeah, very expensive. Now, of course, there's Medicare and Medicaid, but as this article outlines, can only pay 80%. So paying $5,600 a year for a treatment out of not 5,600 actually, 11,200 out of pocket costs for a patient. That's a lot of money, especially for one drug which we outlined has, perhaps high risk of symptoms and might not even have demonstrable clinical benefits. So that's fairly, especially integrated, that is an ethical concern. So billions of dollars of an increase in overall Medicare spending, which, there's lots of patients with Alzheimer's, and even though not all patients with Alzheimer's would be treated with Aducanumab for sure, because, it applies only specifically, as Biogen has stated and the use label has stated, it only applies specifically to patients who have amyloid deposition, which is not every Alzheimer's patient, and also would apply to patients who perhaps don't have the APOE4 gene, and also are early onset. So that's the target demographic. It's not the entirety of the Alzheimer's demographic, but still a lot of patients, a lot of money for a lot of treatment. And that's a burden on the taxpayer. That's a burden on the patient who will have to pay some out-of-pocket costs depending on their level of insurance. Not great from an ethical standpoint when we're talking about the money. So yeah, beneficence. That's talking about the clinical trial data, which again, it was not conclusive that this is a drug that actually slows down the onset of symptoms. So yeah, fairly good beneficence argument to be made there. Won't cure AD, restore cognitive function. I mean, that was from the early days of these antibodies. It was an accepted fact that this is not a curative or restorative treatment, rather that it's a treatment that slows decline, or at least that's what it's purported to do. So not new information, but still it's relevant when you're making a decision, weighing up economics, weighing up risk of side effects. This isn't something that's going to cure, this isn't something that's going to even prevent onset, this is just slowing down. You're just giving the patient a few more years, perhaps, of cognitive function at a very best case scenario. So this is something to consider. Non-maleficence, so this is going to be talking about brain inflammation, where you've got the related imaging abnormalities, and you're going to have, and of course, ARIA-H, we've got brain hemorrhage, ARIA-E, we've got brain edema, or a combination. So the way this study is interpreted the data, more than 1/3 of patients receiving the approved dose of aducanumab, which is of course the high dose because that's what Biogen purported is an efficacious treatment. 1/3 of them are getting these symptoms, which again doesn't necessarily mean 1/3 of patients are receiving or are experiencing direct and provable harm, and as part of the treatment label, regular PET scans are mandatory. So if there were these abnormalities, a clinician could come to a decision to lower the dose or halt the dose for a period of time and then restart it later. So this, it's not quite the same as saying 1 in 3 patients is going to have a severe reaction. That's not, that would be a very different thing. And it says here, 1 in 10 patients actually had headaches, confusion, dizziness, vision changes, nausea, and vomiting that could be directly linked to the full dose aducanumab, perhaps. But the side effects. It does point out the FDA label calls for less frequent monitoring that then was present in the clinical trials. So something to note. But this is an important thing to discuss, the non-maleficence, that there are these symptoms that can be damaging. And again, as we saw earlier in the Reuters article, one patient did indeed die. So, and that does weigh on the expense, because if you're paying money for a treatment that could give you a serious side effect, that is, there are considerations around Hippocrates' oath and, the duty of a healthcare provider to take into account the health of a patient and to treat them justly and ethically. So yeah, this article makes the point that neurologists, if prescribing aducanumab, would indeed need to discuss this with families, especially ApoE4 gene carriers, because that increases the risk, that increases the burden. Justice. So this is going to talk to the earlier concerns I raised. I pointed out from the data around the ethnicities, because genetic disparities indeed can be important for outcomes and treatments. The difference is, for example, between males and females in how they respond to specific treatments have been a major source of concern in terms of justice over the last half decade for medications, because most trials in the very early days were based very much on men or males. So this is an important consideration. If there are very slight genetic variations in different ethnic groups, then if that ethnic group is not properly represented in the clinical trial, then perhaps if a clinician were to refer or prescribe them rather this drug, then there's a conflict in the principle of justice there. And so, if we're going to talk about very slight genetic disparities, genetic differences, and ethnicities, that can have a fairly high impact when it comes to clinical outcomes. We can just talk about the very recent and very prominent SARS-CoV-2 epidemic, with the fact that Asian people of Asian descent tended to have a higher amount of ACE receptors on their lung epithelial cells, and ACE receptors were the receptors that SARS-CoV-2 molecules entered, which made them more susceptible to the disease. So that's an important point there. So yeah, talking about out of course, it is significant again to note that the taxpayer who's not taxpayers are of an ethnicity that's not directly treated by this drug, they're paying money into covering the cost of this treatment, but it's not been proven to directly benefit, that it could benefit them. So that's a concern. Fewer than 3% of the participants enrolled were Hispanic and Black and Indigenous, which is pretty. So there's a pretty significant element to this, that The drug's not been EMA approved, so in Europe there's a there is a far higher percentage of white and Asian ethnicities as a group of the population compared to America, which is with the FDA is approved, which actually is a much higher proportion of Hispanic and black ethnicities as opposed to Denmark and Sweden and the UK, which were countries that were and Australia, countries that were included in the studies. So yeah, that's a key point. Autonomy. So this will be talking about what an individual patient wants. So we'll just skip to the key points. Respect for patient autonomy. So the clinicians will have to make fair representations of this treatment to patients. And it does mention that for some hospitals there are financial incentives for clinicians to hand out high-cost drugs. They'll get money back if they prescribe more treatments. So that is an ethical consideration that some clinicians would be financially incentivized to hand out these drugs that perhaps might not be suitable for an individual patient. So that's a consideration, especially if it conflicts with patient autonomy. If can a patient make an informed decision if a clinician is presenting a biased take on the data because he or she might want a certain drug to be administered. So yeah, these are all very valid, very important concerns and considerations. So discussion regarding potential benefits. Again, we're talking about non-maleficence, the absence of convincing evidence for efficacy, talking about potential benefits being modest. We're talking about the risks of edema, hemorrhage, and hospitalization as a result. Talking about burdens of the administration monitoring. There is a financial burden for all these PET scans. That is an additional cost that would be a burden on the healthcare system and perhaps on the patient and family as well. And these are all the broader considerations as mentioned above. So that's the AAN's position statement. And these considerations that we brought up will probably provide focal points for the conclusion of this video. Thank you very much. So that's the video finished. It took a lot longer than I originally anticipated, but I hope I've been able to go through all the data, not all of the data, but go through a significant representation and proportion of the data, so I've been able to introduce to you and explain a fairly comprehensive analysis of this topic, of the topic of Aducanumab and whether it is actually safe and actually ethical to be used as a drug, whether the FDA did make a mistake. So I've presented all the data that's requisite for perhaps this discussion and conclusions. So I'm going to make my own subjective conclusion where I'm going to go over the points made by D.Knopman, go over the points made by the AAN, and come to a conclusion which you'll probably be able to anticipate. And then we'll finish this video, so it's probably going to be over an hour long. But if you've stuck with it, I hope you've enjoyed it, and I hope there hasn't been too much repetition or redundancy or umming and ahhing. So yeah, thank you very much for sticking with me this long, and we'll get right into the conclusions. So we have the first consideration, which is in the abstract of the results of Verubecestat and the potential falseness of the amyloidosis hypothesis. So if the amyloidosis hypothesis as it stands today is false, if amyloid beta soluble proteins and their deposits aren't actually a cause for decrease for symptomatic onset of Alzheimer's disease, then that has a very big impact for the entire field of these neurologics, these second-generation Alzheimer's neurologics, which include lecanemab and donanemab and aducanumab and all these. So, that's a pretty big concern. Again, we can't make conclusive judgments there because the data's not all out. The jury is still out on this particular topic of amyloidosis will be falsified or verified. Oh, who knows? We will see. But that is, of course, a consideration, and it appears that if we were going to take the specific example of Aducanumab, then a critical analysis of amyloidosis would be borne out evidentially by this specific example, and also the example of Solanimab and Verubecestat that we looked at earlier. So that is a consideration. But if we're going to look at specifically Aducanumab, and the question which I want to pose in this video, after having looked at in the first video of the proposed mechanism of action of this drug, and having looked today at all the issues and controversy surrounding this drug. So again, the principal question I want to propose is, well, should it have been approved? Should it be a drug that's on the market? Should it be a drug that Alzheimer's patients are receiving? And I think all of you who have listened so far and who have reviewed the data will probably join me in concluding that no, this drug seems to violate principles of non-maleficence, of beneficence, of justice, of ethics. This is, and if you add on top of that the fact that the board, arranged by the FDA, actually 10 stated that clinical outcomes were not proven to be beneficial for this drug. And again, that's not even saying the drug. That's not even saying that the drug could be approved, because that's weighted by symptoms and finance, which could be ameliorated by other means. But that's just the value judgment on the efficacy of the drug itself, and that's a fairly valid judgment. After we looked at the emerge and engage trials, from that data. So that would be disheartening for Biogen, especially after all that money they've put in. But the FDA then decided to fast track this drug for no discernible reason, and it's been approved, and it's now currently on the market. And Biogen states that they are monitoring the drug and monitoring its efficacy now that it's being administered to a wide range of people. But again, this is problematic. And then we'll look specifically, just briefly outline the points that, this is a high financial burden on both the taxpayer and perhaps the individual patients paying out-of-pocket costs for this drug. This is significant risk of side effects, and again, as you'll remember, 1 in 10 of the patients who as part of the trials had severe symptoms associated with the amyloid imaging abnormalities. So that's a concern, especially since the Reuters article, remember earlier, the one patient that died, one patient, but still, that's a marker perhaps of a potential harm, which, if you expound that across a whole population, especially since the mechanism for catching these severe symptoms, these side effects, before they developed into a serious condition, would be PET scans for these RIH and RIEs, but in the medical system, PET scans will be far reduced as opposed to what Biogen provisioned in their clinical trials. So that's an important concern, the maleficence concern. And then the justice concern, the fact that, this drug only has proven, even if there's proven efficacy from the clinical trials, it only has that proven efficacy for the white and Asian ethnicities that made up the trial pretty much in its entirety, which, as anyone watching this video will know, is not representative of America. There is, not everyone in America is white and or Asian by ethnicity. So, these are all significant concerns. And again, I will reiterate my personal conclusion, which is that Aducanumab should not have been approved. It should not be approved now. More data will come out as to the implications of its efficacy. So when we see patients, after it's been around 2 years since it's entered the market, we should start seeing fairly large groups of patients, and the benefits or perhaps harms that have been brought upon them. So that data is going to come out, and perhaps I'll do a video on it another time. But that being said, I think the data that we've seen and the evidence that's been shown doesn't provide a justification for Aducanumab. There should at least, as according to David Knopman's recommendation, been another phase 3 clinical trial. And while it is understandable why Biogen wouldn't want to do that, that's a lot of money. Again, it's not excusable. These are people's lives and livelihoods. So that's the conclusion that I've come to. Now, I hope all of you can look at the data yourself if you're interested in the topic. There are many videos that have more colourful and bright and interesting, perhaps, descriptions of this amyloidosis hypothesis, of perhaps the drug, and perhaps considerations around the FDA and around regulation, all these different things that are very briefly touched upon in this video, and I will provide links for further reading if anyone's interested. But yeah, I hope all of you enjoyed, and hopefully it won't be so long before my next video. I'll try and make this one a bit less niche, a bit less niche, and I'll try and have it out for you guys sooner. But yeah, thank you very much for watching. If you can, please, if you want to, please like, subscribe, and share the channel with your friends if you think they would be interested in this content. And all of you who are studying in the biomedical field and are interested, just go for it. You guys got it. And if anyone I know is watching, don't mark me too hard. Right. Thank you very much, and I'll see you all another time.